Hello, how are you? Do you hear me? Good, thank you. Great. So hope some other students will join us, but we can start already. So last time we couldn't manage to cover pathogenesis and antimicrobial chemotherapy points, yeah, important topics, because so this first lecture in bacteriology was quite big, covering many topics. So we can start with, we had just partly covered pathogenesis as I remember, so we can start today. From pathogenesis, we'll cover antimicrobial chemotherapy, also sterilization, disinfection, antiseptics. Also, we'll briefly discuss diagnostic methods in microbiology. Yeah. Do you have any questions? Oh, firstly, how was the midterm? We did not meet, I think, after midterm. Yeah, okay, good. So it wasn't difficult, yeah? And time was appropriate, not too short. I have asked it to be 1 hour. Was it too long or was it okay? What do you think? Okay, for the next midterm, let's keep the same time and same number of questions. Yeah, so for the second midterm, you will have again 30 questions and 1 hour. And I have discussed the time of the second midterm, so it can be in week 12. Yeah, 12, yeah, after how many? Yeah, I think it's okay. Yeah. Um, okay, great. So, do you have any questions regarding our course or some specific topics? 2 more weeks or even more. Yeah. Today is this week is 8th week, I guess, if I'm not mistaken. We will finish our semester in July. We have 15 weeks in total, yeah. Now is eighth, yeah, eighth week. So another 7 weeks. Maybe somewhere, yeah, in the middle of July. It should be indicated in portal. This information should be provided on a portal, the exact date, but approximately it will be, I think, around 10th, July 4th. Yeah, okay. Okay, so maybe by that we'll finish our course. Yeah, I hope so. We'll see. 5 more weeks, but we have 7 topics left. Maybe it's old information that the end of semester is 4th of July. I don't know. I have to check because 7 weeks are left. I think we'll finish by 15th or I don't know, 10th of July. Maybe more by 15th. So we'll see. Okay, and you will have midterm after 4 weeks, the second midterm. Um, I don't know, maybe online, yeah. I think so. Yeah, this whole semester will be online. I think finals will be also online. Yeah, because all students are in, so majority of students are not in Georgia. Yeah, so it's better if you stay in your homeland and if there will be online final exam and starting from the next semester you will arrive again. I hope will not have some serious waves of corona in autumn. Anyway, in beginning of September, September, October shouldn't be the problem, I think. Maybe a second wave will start in November, second part of November. At least part of the semester will be in Georgia and not online. So we'll see, yeah? We don't know what will happen in the future. Okay, so let's start our tutorial and let's discuss pathogenesis first. And I want to ask you, how do you understand this pathogenesis? Have we discussed this? I don't know. I have so many courses. I already forgot. I don't think we haven't discussed what is the meaning of pathogenesis. Yeah, with you. How do you understand this pathogenesis of bacterial infection, for example? What do you think? What is pathogenesis? So we're going to discuss pathogenesis. And so what's pathogenesis? I will open my PowerPoint also in my mobile, my presentation. Development of a disease. Yes, you're right. And yeah, mechanisms, yeah, by which the disease develops. Yeah, and in pathogenesis, we may have the role of bacteria, we may have the role of immune system, yeah? So pathogenesis means that pathogenesis implies or means mechanism by which the disease develops, yeah? Great. What about virulence? What is the virulence? What virulent? Firstly, let's discuss what is pathogenic bacteria. Which type of bacteria are pathogenic? Yeah, you have heard, yeah, pathogenic bacteria, pathogenic viruses. Which are these pathogenic bacteria? I mean, yes, I'm not asking about genera, but cause disease. Yes, exactly, Hussein. Thank you. So, pathogenic bacteria are the bacteria that are able to cause the disease. Yeah. And what about virulence? Which are the virulent microorganisms? We have here highly virulent, not so virulent, yeah? Poisonous virulence is your close, of course. Virulence means measures is actually one qualitative measure of pathogenicity. Yeah, for example, one type of bacterium, for example, Salmonella typhi, that causes typhoid fever. Yeah, it depends on severity of a disease, but yeah, the disease severity can depend on virulence of the strain. Yeah, how virulent is the strain? For example, Salmonella is the pathogenic bacterium, but some strains of Salmonella typhi that causes, for example, typhoid fever, this is a very severe disease, are highly virulent. Some strains can be of low virulence. Yeah, highly virulent strains can cause severe disease, while the strains with the lower virulence have a not so good ability to cause the disease. Yeah, and the virulence is determined by the ability of the microorganism to produce toxins, enzymes, some factors that help the bacterium to evade immune system. Yeah, we have talked last time about Streptococcus pneumonia that forms capsule. Yeah, some strains may not cause form capsule, and they are less virulent. Yeah, their ability to cause the disease is low. The species Streptococcus pneumonia, yeah, is pathogenic organism. So in general, this species causes the disease. Yeah, but under this species, at subspecies level, yes, strains can differ. Yeah. So some are more virulent, others are less virulent, and of course, it largely depends on the genetic properties. If the bacterium has virulent genes that encode toxins, in this case capsule, yeah, so we have low and highly virulent strains. So this is virulence. This is actually qualitative measure of pathogenicity. So how good is the bacterium to cause the disease? Yeah, how strong is the bacterium to cause the disease? Does it make sense? Okay, great. And now let's move. I will share now with you my presentation, and we can go through the slides. So, here we have definition of pathogenesis again on this slide. And we have types of infection here, yeah? So I will move to our meeting window. So, and I wanted to ask you if you understand the meanings of those types of infections. Yeah? So we have here bacteria can cause opportunistic infections. Yeah? What is opportunistic infection? Do you understand this meaning of opportunistic? Do you know the meaning of opportunistic infection? So, great, Ali. So, what's the opportunistic infection? Yes, infection caused by pathogen, but not really pathogen. Opportunistic infections can be caused by non-pathogenic or opportunistic microorganisms. They are not strictly determined as pathogenic. Opportunistic means that if we're healthy, usually many bacteria don't cause disease, yeah, But if we get immune suppressed, many microorganisms can lead to the disease development. These are opportunistic infections. Yeah, for example, in one of our lectures, we have mentioned our microbiome, yeah, microflora, where we have many hundreds of thousands of species of bacteria as well as viruses. And those bacteria usually do not cause the disease, yeah, and they are beneficial. For example, can be E. coli, lactobacilli, bacteroides, so many aerobic or facultative anaerobic microorganisms live in our GI tract. But sometimes during surgery, those microorganisms can contaminate the area of surgery, yeah, so wound, and can cause the infection, yeah. So under natural conditions, one person is healthy, no infection with these microorganisms, but under certain conditions, yeah, those microorganisms can cause the disease. These are opportunistic microorganisms. And I can give you also other examples of immunosuppression. Yeah, AIDS patients, so patients who are infected with HIV and who have progressed, who have disease, yeah, already, they show many opportunistic infections. Yeah, they can be infected with the bacteria that are present in the environment, in the soil, Mycobacterium avium complex, for example, that usually doesn't cause disease in healthy people, but in HIV patients, it's a problem. It may cause tuberculosis-like disease. Also, many pneumocystis carini, this is fungus that in healthy people don't cause disease, but in AIDS patients, yes. So all these microorganisms are opportunistic pathogens, and so these type of infections are opportunistic infections. Does it make sense? Great. So, what is a primary infection and what is a secondary infection? We have also here acute, chronic, subclinical, latent, localized, generalized, pyogenic, polymicrobial infections. Let's go. It's important for you to know all these types of infections. What is primary infection? Yeah. So first time to be exposed, this is primary infection, and secondary, consequently, is secondary may have two meanings. So what do you think? Secondary infection. Secondary, not really after treatment, but classic secondary infection develops, for example, after it can be caused by the primary. Sometimes secondary is secondary. Yeah, the classic description of secondary infection is when I was infected with one pathogen, and this first pathogen then helped other pathogens to cause the disease. I will give you the example. When we have the flu, yeah, flu actually disrupts the ciliary action of our ciliated membrane, not membrane, the ciliated cells of the upper and lower respiratory tract. So it destroys, flu virus destroys those cells, ciliated cells, and we become more susceptible to bacterial infections. Yeah, so we have discussed in immunology, yeah, how important are these ciliated cells of respiratory tract. Yeah, so they remove microorganisms as well as foreign particles. So if flu disrupts and destroys those ciliated cells, yeah, so we are more susceptible and we can get infected with staphylococcus, haemophilus, or so with streptococcus pneumoniae again. So those are secondary infections. Yeah, and so I had some infection, and then this primary infection reduced my immune response or something, and then some other bacterial or can be viral infections that appear. Yeah. Also, when people have children chicken pox, yeah, this is a rash, vesicular rash on the skin, and the skin is damaged. Yeah, this is viral infection, but as the skin is damaged, again, bacterial infections can develop at the site of damage, yeah, skin damage. At the where we have vesicles, there's a skin damage and bacterial infections develop. These are also examples of secondary bacterial infections. Yeah, that's why people who have chickenpox use some antiseptics. Yeah, can be brilliant green or iodine or something, yeah, to prevent secondary bacterial infections. Does it make sense now? Not after treatment. There can be no treatment. For example, may not treat a flu at all. Yeah, we are in chickenpox. There are treatment options for flu, but we don't use it in all cases. Yeah, and in chickenpox, if there's we have a cyclovir, this antiviral medication, but it's not so commonly used. Yeah, we may not use any treatment, but after a primary infection, yeah, secondary infections may appear, depending what damage is done by the primary infection. Yeah. Okay. Now, what is now acute infection? Mild, yes, usually acute infection. Short period, Hussein, yes, thank you. We have short period of, yeah, relatively these acute infections are usually mild, yeah. Yeah, symptoms can be, yeah, symptoms reach the peak and then the symptoms disappear. Depending on infection, yeah, it can be, symptoms can be either severe or mild. Can you give me examples of acute infection? From your experience, yeah? Any infection that resolves within several days. Flu, yes, it's exactly. Fever is one of the symptoms. Yeah, yes, some febrile illnesses, yeah, flu, or non-febrile illnesses, for example, mild gastroenteritis or severe gastroenteritis are also acute infections. Yeah, caused either by viruses or bacteria. Yeah, so in acute infections, we have short incubation period, and pneumonia. Yes, yeah, can be acute infection, result of an acute infection. Yeah, uh, and yeah, so we have incubation period, then prodromal period, then we have symptom development, then symptoms disappear and we recover. And acute infection can last for a few hours, several days, several weeks, a few months, because hepatitis can last for several weeks, but it can be acute, depending which hepatitis virus causes that. But what about chronic infection? Yeah, it lasts for a long time, Hussein. Thank you. Can be years, yeah, like HIV. Yes, Alexia, symptoms develop for a long time, and we have persistent, long-lasting infection. Yeah, can last for years. What about subclinical? How do you understand this? Subclinical infections. Asymptomatic, yes, that's great. Thank you, Ali. This asymptomatic. So subclinical, sub means usually under, yeah, under clinical presentation, subclinical. What about latent? Well, latent infections can, at some point, are um, may have no clinical presentation, but they can reactivate time after time. This latent, for example, herpes simplex virus infection, when people have fever blisters around the nose or around the mouth. Yeah, this is latent reactivate. It's also a form of the chronic infection. We have lifelong infection once we're, yeah, like hidden. Yes. So virus is hidden, you're absolutely right, and then can reactivate time after time. And latency is in neurons of HSP. What about a localized infection or generalized infections? What is localized infection? So name indicates this is localized, yeah? Yeah, it's a texture specific, yeah, part of the body. Yes, you're right. And usually this part of the body is portal of entry. Yeah, for respiratory viruses, this can be upper respiratory tract. Yeah, and those viruses may not spread. Yeah, this is localized. If we have wound infection caused by Pseudomonas aeruginosa, this is also localized skin infection. If a virus or bacterium or fungus do not spread through blood or lymphatics or through neurons, yeah, we have localized infections. So consequently, which type of infection is generalized? Or systemic? The opposite, yeah. So bacterium or virus spreads and can affect different parts of the body, can spread to different organs, liver, spleen, brain. A fever is can be a result of localized infection as well. Yeah, not of systemic spread. Localized infections like flu also causes localized infection. We don't have the systemic spread. The virus doesn't go to the blood and very rarely, so it is an example of localized infection, but we still have fever there. Also, some viruses that cause gastroenteritis or some bacteria For example, Shigella, dysentery causes very severe gastroenteritis, but it's like localized infection. Bacterium does not spread beyond the GI tract, but there can be fever and bloody diarrhea. Yeah, so it's the toxins may have systemic effect. Yeah, produced by the bacteria that cause localized infections. Yeah, but also viruses that cause localized infections. Usually, viruses don't produce toxins. There are a few exceptions. But they can induce systemic effect, yeah, fever and so on, myalgia, headache, yeah. But it's not a systemic infection or generalized infection. Okay, what is a pyogenic infection? Produces pus, yes, thank you. So pyogenic staphylococcal infection of the skin, yeah, when pimples develop, or propion bacterium or some other that causes pus formation, yeah. And polymicrobial infections, the name indicates, yeah. So what does it mean? Poly means many, yeah. Polymer, yes. Yeah, thank you, Ali. It is caused by many bacteria. So, periodontal disease, also caries for dentistry. Yeah, the caries mainly is caused by Streptococcus mutans, but other bacteria like lactobacilli can help, and also others. Periodontal disease is mainly caused by Porphyromonas, and bacteroides can be also involved, and other anaerobic bacteria. Yeah, so these are examples of polymicrobial infections. Okay, let's move to the next slide. And we have here a description and that bacteria cause disease by two major mechanisms, yeah? Invasion and inflammation, and also by toxin production. Yeah, so it is, I think, easy to understand. So either bacteria directly attack the cells and tissues, yeah. So they infect those cells, or can be extracellular bacteria, but they can make the damage to the tissue and cause the inflammation, or toxins cause the disease. Yeah, so toxic, I can give you examples of diseases that are caused by preformed toxins. We'll learn this in the following lectures, of course. And for example, staphylococcal toxin can cause poisoning. So, but this is preformed toxin. Staphylococcus is in some food. Yeah, staphylococcus grows and produces toxin. And if we heat this dish, Staphylococcus will die, but toxin is heat stable. We will eat this dish and will get food poisoning. Yeah, and there are many examples. Vibrio cholerae actually causes disease due to toxin production. Also, Clostridium botulinum, botulism, is caused by toxin production. Tetanus, yeah, also through toxin production. Yeah, either we have direct invasion of tissues and cells by bacteria and inflammation there, or microorganisms produce toxins, and the disease is the result of the toxin action. Yeah. So let's discuss. Toxins are very important mechanisms of pathogenicity. Yeah. And here we have some toxins, exotoxins. We have discussed briefly rather last time, yeah, endotoxins and exotoxins. Could you tell me which are the endotoxins, which is in, yeah, what is endotoxin and which are exotoxins, yeah? Yes, and from inside, not of our body, but gram negatives. Who's saying yes? Thank you. Gram negatives have endotoxins. This is part of the lipopolysaccharide. Lipid A, yeah, is a endotoxin, while exotoxin is excreted, or what happens here? So exome is outside the cell, yeah. So bacteria excrete this from the cells, yeah, and they are products of bacterial metabolism, and they go outside the cell and have this toxic effect. And we have examples of some toxins here: leukocidin, streptolizin, botulinum toxin, diphtheria toxin, tetanus, cholera toxin. So we will discuss. These are all, majority of them are exotoxins, yeah, produced by the bacteria, and they play important role in pathogenesis of bacterial infections. And exotoxins are secreted, yeah, from bacteria, and bacteria have different types of secretion systems. And some of the so there's one type of secretion system like a syringe. Yeah, this is type T3 secretion system. Okay, let's move to the next slide. So I'm sure you all know the mechanisms by which the microorganisms are transmitted. We have mentioned, I think, already, yeah, respiratory, fecal oral route, skin, genital tract. So mainly microorganisms enter our body through skin or mucous membrane, yeah. And what other route of transmission is missing here? Something is missing. What do you think? Which route of transmission or several routes are missing here? Main routes are mentioned, but we can get microbes through other routes as well. Can you guess which are the other routes? So here, respiratory, fecal oral, skin, genital tract. Yes, okay. But we can get microorganisms through other routes as well. Any idea? What about blood transfusion? Maybe you have heard, yeah? So the blood is checked for hepatitis, for HIV, for different microorganisms. Why? Because one of the routes of transmission can be blood transfusion, yeah? Also, surgical manipulations, yeah, in dentistry also if one is not sterilizing the instruments well, yeah, so it is another route of transmission, yeah. Yes, yeah, so medical manipulations in general, injections, blood transfusions, yeah, so these are invasive techniques, yeah. But some arthropods can also transmit infections, yeah, insects or ticks. Yeah, so we have some arthropod-borne diseases. Many viral encephalitis or hemorrhagic fevers are transmitted by vectors, by insects. Yeah, many parasitic infections can be transmitted by insects. Tsetse fly in Africa. Have you heard about these? Transmits African sleeping sickness, for example. Yeah, so Insects can also transmit. And one major route is missing here. This is vertical transmission from mother to child. Yeah, transplacental transmission. So some viruses, HIV, measles, rubella, cytomegalovirus, parvovirus, among protozoans, toxoplasma can pass placenta. Um, the bacterium that causes syphilis, Treponema pallidum, can cross placenta. Yeah, so different bacterial, protozoan, viral infections can be transmitted from mother to child, and this transmission can occur during the pregnancy. Yeah, intrauterine transmission can be during the birth. Yeah, during the breastfeeding as well. Yeah, so we have here different routes from mother to child transmission. Okay, and so second point here discussed is that bacteria, attachment of bacteria to the surfaces is important for initiation of infection, of course. Here we have importance of biofilm formation. Yeah, last time we have discussed biofilm formation. Dental plaque was a good example of biofilm. Now, and when we were talking about capsules and the slime layer, yeah, bacteria produce these slimy things, and then other bacteria come, stick together, and then they form the biofilm, and they are very well protected from antimicrobial agents and bacteriophages as well. Yeah, so biofilms are very important. Yeah, and let's also discuss the enzymes. Yeah, so enzymes, bacteria produce different enzymes. Why? What do they need? So, why enzymes are important, and what are the functions of enzymes produced by the pathogenic bacteria? They can produce different types of enzymes. Can you give me examples of those enzymes and what are their function? What is the function of those enzymes? Yes, to protect them, yes. Yes, this is the not only protect, yes, one function can be this, major goal can be this. Yeah, to protect, yeah, to overcome body defenses, yes, because some produce enzymes, yeah, but major, yeah, lecitinase, for example, lecitinase is the enzyme that can kill blood, different types of cells in the blood, yeah? Yeah, red blood cells, yes, yes, right. There are also, and yes, you can think and tell me, there's enzymes that are important for spreading and for invasion. Can you give me examples? Siderophores, yeah, they are, what are siderophores? What is their function? Yes, iron capturing. Yes, Alexi and Hussein, thank you. Yeah, so iron is always fixed in our body. Yeah, we have lactoferrin, hemoglobin. Yeah, so iron is captured, and they have pathogenic bacteria have siderophores that have very high affinity. Yeah. Very high affinity to iron, and they capture this. Yeah. And bacteria have also hyaluronidase. Yes, they can have coagulase, different enzymes that enable them to spread collagenase, yeah, so hyaluronic acid and collagen is a major component of connective tissue, so they hydrolyze and can spread easily, yeah, through by presence and by secretion of those enzymes, so they can easily spread in our body. Yeah? Okay, now we have here the action of exotoxins. Yeah, in general, mechanism of action. Yeah, and as you can see, we in exotoxins we have usually two subunits. Yeah, there are some exotoxins that have more subunits, but in general, exotoxins have two subunits, A and B. Yeah, so as described here, and B subunit is for attachment, and A for attachment and for transportation of A subunit inside the cell for endocytosis, while A has the toxic action. Yeah, so this is AB toxin action. Yeah, so B attaches to the receptor, then we have endocytosis of the exotoxin, then A and B subunits are separated, and A can have the action like to block the protein synthesis, to block action of the elongation factor 2, for example, in some bacteria. Some can for even tetanus or botulism, we have different type of action. They block uh, release of acetylcholine. Yeah, for example, this is possible. But so different toxins have different mechanisms of action. In cholera, this toxin induces the leakage of the electrolytes, ions, and water into the lumen, for example. Yeah, so different toxins can have different actions, but all of them are AB toxins. Yeah, so they have two subunits. Exotoxins have mainly two subunits, but there are exceptions, as mentioned. And of course, we'll discuss these toxins when talking about each bacterial species. For example, Corynebacterium diphtheria produces exotoxins. Yeah, also Clostridium tetani, Clostridium botulinum. Yeah, so we'll discuss this in more detail when talking about these microorganisms. And I did not include in the lecture Koch's postulates, but I want to describe briefly what is this, as it is also described in your handbook of dentistry, microbiology for dentists, for dentists, yeah. So what is this Koch? Koch was the guy, German guy, very famous microbiologist who discovered Mycobacterium tuberculosis, is Koch's bacillus, yeah, the same as Koch's rod. So Mycobacterium tuberculosis, bacterium that causes tuberculosis. He worked on Bacillus anthracis, the bacterium that causes anthrax. Yeah, so he was a very, very important guy, and he first introduced the cultivation of bacteria under laboratory conditions. He first developed the culture media, yeah, and solid agar media and so on, yeah. So, what are these Koch's postulates? The first postulate is that no. So, what is the aim in general? Cross postulate is how to prove that certain microorganism causes the disease. Yeah, how can we prove this? If the organism or animal or human is sick, yeah, we should be able to observe this microorganism in the body. We should check and we may find this microorganism, can be a microscopy or whatever. So the causative agent should be present in the body, yeah, of the diseased individual, but should be absent in healthy animal or human. Yeah, we should be able. The second postulate. So we have one, two, three, four. Yeah, there are four postulates. And second postulate states that we should be able to isolate the causative agent from the diseased individual and culture under laboratory conditions. You see the second. Yes, the causative agent must be isolated and so on. Yeah. And if we inject this isolated, for example, bacterium in the healthy mouse, in this case, this bacterium should cause the same disease with the same symptoms. Yeah. And from the infected mouse, again, artificially infected mouse, we should be able to isolate the same bacteria in the pure culture. These are four postulates. It was uh, the Koch wrote in 19th century, but of course it is still valid. But many bacteria are non-culturable, yeah, so they may not meet Koch's postulates, but we have new diagnostic tools to prove that certain microorganism causes the disease. Though the viruses, for example, many viruses are not culturable, yeah, we can see and we can detect those viruses in diseased person by immunologic methods or by PCR genetic or molecular biological methods. Yeah, so we can use other diagnostic tools. So the new approaches, new techniques can fulfill this postulate. Yeah, does it make sense or is it difficult for you to understand? Hello. Yes, clear. Thank you, Ali. Okay, let's move to MCQs now. So, which of the following statements about lipopolysaccharide is correct? It interacts with the macrophages and monocytes, yielding release of cytokines. The toxic component is the O side chain. It forms holes in red blood cell membranes, yielding hemolysis. It causes hypothermia, it causes paralysis. So it's simple, I guess, yeah? What do you think? So lipopolysaccharide, this is, it includes endotoxin, yeah? One component is endotoxin. We have lipopolysaccharide B. Toxin component is the O side chain. Not O side chain, but we have lipid A here as a toxic component in lipopolysaccharide. Yeah, O side chain is variable chain and is used for serotyping for different bacteria, different gram-negative bacteria. But toxic component is here the lipid A. And so lipopolysaccharide is the component that is the structural component of the bacterium that can lead to fever, inflammation. So which option is correct here? What do you think? D, not hypothermia. Hypo is low temperature, yeah, not increased temperature. But how this fever is induced, yeah, macrophages and monocytes, actually macrophages engulf gram negatives, yeah, so they digest, and then different sites. So it's A, yes, I'm saying it's A. It interacts with macrophages, and so we have here release of cytokines, then prostaglandins and so on, yeah, leukotrienes, and we have inflammation and systemic. Okay. Which of the following statements is correct? Maybe it is difficult now for you to answer these questions because we had just a general overview, but these questions are related to pathogenesis. If it's difficult for you, I can help you. Yeah, of course. Yeah, in general descriptions, these specific microorganisms are also mentioned as an example. Yeah, that's why these MCQ questions, review questions of a general part, can be at some point specific to certain organisms. Yeah, because we see examples, but I don't think that it's important now to memorize this, which toxin is produced by which bacterium also, in general part, because later we will learn this step by step. Yeah. So I will help you here. Which of the following statements is correct? Lipopolysaccharide is part of the cell wall of E. coli. E. coli is the gram-negative bacterium. So what do you think? Should it have lipopolysaccharide or not? Yes, so A is correct. Yeah? Cholera toxin is attached to the flagella of Vibrio cholerae, and I have mentioned this cholera toxin is exotoxin, so it cannot be attached to the main component of the cell. Yeah, so it is released. The B is false. The lecithinase or Clostridium perfringens causes diarrhea. This you may not know, but lecithinase degrades or causes lysis of red blood cells, for example. Toxic shock syndrome toxin 1 is produced by hemolytic strains of S. epidermidis, Staphylococcus epidermidis. Actually, Staphylococcus aureus produces this. So, well, next topic, what we will have will be Staphylococcus and Streptococcus, and we'll learn this in more detail. So now let's discuss mechanisms of action of antibacterial drugs, antibacterial chemotherapy, yeah? And so this slide describes very well what can be the targets of the antibacterial chemotherapeutic agents. Yeah. Firstly, how do you understand this term selective toxicity? So, when we are talking about medications used for treatment or development of medications, we very often talk about selective toxicity of those medications. What do you think? What is that? Selective toxicity. Selective toxicity. Yeah. Yes, Hussein, thank you. It should be specific to target microorganisms and should not affect the host. Yeah, it is important. It should be selectively toxic to the target, but not to the other cells, especially to the host cells. Yeah, and this is important factor in selecting the medications for treatment. So, as you see here, we should find something in bacteria that is bacterium specific. Yeah. So, for the target for antimicrobials. Yeah. For example, target of medication of antibiotics should be something that only bacteria have and not the host. Yeah. That's why. And what are these components? Is the cell wall specific for bacteria and not for us, for example? Do we have peptidoglycan, for example, in our cells? Or is it bacterium specific? Bacteria specific, yeah. Uh, Peptidoglycan, no. If so can you imagine we have peptidoglycan as well in our cell surface, yeah? And on this slide, you see that some antibiotics, for example, beta-lactams, vancomycin, bacitracin, also cephalosporins, so penicillin, so beta-lactams are those antibiotics, attack cell walls of bacteria. They actually block peptidoglycan synthesis. Yeah, if we would have had this peptidoglycan, of course, it would be toxic to us as well. Yeah. So, why cell wall is considered as a target of antimicrobials? Because peptidoglycan is present only in prokaryotes, in bacteria, but not in eukaryotes. That's why we use medication that targets cell wall, and that's why these antibiotics are, of course, antibiotics have some side effects, but of course they're not very toxic to humans because we don't have the target. We don't have peptidoglycan. Does it make sense? Okay, great. So we have one of the major targets of antibiotics, antibacterial medications is to block synthesis of peptidoglycan. And you see very well-known antibiotics here, penicillins, for example, beta-lactams. Beta-lactam is uh, lactam ring is chemical structure, yeah, in the molecule. That's why beta-lactams. And penicillins, cephalosporins, carbapenems, monobactams belong to those group, yeah, that group, beta-lactams, vancomycin, bacitracin, yeah. So these antibiotics attack the cell wall and block the cell wall synthesis, yeah. And bacteria may die or may stop growth, yeah. Um, and what can be other targets? Yeah, we have here a nucleic blockage of nucleic acid synthesis, either DNA synthesis or RNA synthesis. Yeah, you see here quinolones, for example. Have you heard these antibiotics? Ciprofloxacin, yeah, very broad range. Broad range, what does it mean? Broad and narrow range. antimicrobials. How do you understand this? Which antimicrobial has, I'm not asking for the name, but how do you understand this broad range or broad spectrum or narrow range and narrow spectrum of action? Broad, you're close, thank you, Ali. Broad means that target is the same, can be the same. For example, blockage of DNA synthesis by blocking DNA gyrase enzyme, but this antibiotic can kill different genera or many species of bacteria, both gram negatives or gram positives. So this antimicrobial is effective against broad range of bacteria. Yeah, many different types of bacteria. While some antibiotics may have a lower range, host range, not host range, but activity range. Yeah, so some antibiotics can be effective only against gram positives, for example, yeah, and they have narrow range. While this ciprofloxacin can be effective against gram positives or gram negatives, yeah. Um, so narrow and broad. So in broad range, we have many bacteria. Yeah, so medication targets many bacteria in narrow, smaller amount of bacteria. Yeah, so number, sorry, not less species or general bacteria are affected by narrow range antibiotics. So we have here also, so we have cell wall synthesis as a target, we have RNA or DNA synthesis as a target for antimicrobials, we have protein synthesis as a target, and you see different antimicrobials can bind either to 30S or 50S subunits of ribosome and can block the protein synthesis, yeah, translation stage. Tetracyclines, for example, aminoglycosides, yeah, they block protein synthesis, macrolides, and so on, chlorophenol, and so on. And some antibiotics target cell membrane and they disrupt the cell membrane, increase the permeability of the cell membrane, so the cell dies, yeah? And we have also another mechanism. This is interference with the metabolic pathways in the bacteria. This is, for example, folate synthesis, as you see yeah, on this slide. And synthesis of folic acid, this is important step before DNA synthesis starts, is blocked by sulfonamides, and trimethoprim also blocks, so different steps in folic acid synthesis is blocked. So very often trimethoprim is used together with a sulfametaxazole, yeah, combination. It's used for treatment of many gram-negative bacteria, for example. So it is also widely used antibiotics. Biceptol is the commercial name, for example, of this trimethoprim sulfametaxazole. Okay, so remember, major mechanisms are cell wall synthesis, how these antimicrobials act. They either block the cell wall synthesis, they block nucleic acid synthesis, they can block protein synthesis in bacteria, they can interfere with the metabolic pathways, for example, synthesis of folic acid. They can disrupt, they can form pores in the cell membrane and the cells rupture. Yeah, so these are the major mechanisms how antimicrobials work. And have you heard about antibiotic resistance problem? So we have the world has nowadays quite huge problem of antibiotic resistance. Yeah. Uh, and what do you think? How bacteria can develop the resistance to antibiotics? He is written, but still, what can be the mechanism? Yeah. Have you heard about this problem, antibiotic resistance? Yeah, okay. So bacteria have developed that cannot be treated with antibiotics. Yeah, so it's a huge problem. And especially in hospitals, nosocomial infections. I don't know, maybe you have heard about this, nosocomial infections, so hospital-acquired infections. All hospital, majority of hospital-acquired infections are caused by antibiotic resistant blocks the treatment, but what's the mechanism? So bacteria, they grow very rapidly and they can mutate, yeah? So when they mutate or change, there's some genetic changes by mutation, they can become resistant to antibiotics. How? They change the targets of the antimicrobial drugs. Yeah? For example, these beta-lactams inhibit cell wall synthesis by inhibiting transpeptidase. This is enzyme that actually forms um, cross links between peptidoglycans. So this is enzyme transpeptidase. This is important for cell wall synthesis. And beta-lactams target this transpeptidase. Yeah. And what bacteria does then? Through mutation, this transpeptidase changes so that those beta-lactams can no longer inactivate this transpeptidase. Yeah, so bacteria start to produce different transpeptidase and cell wall synthesis is not blocked. Yeah. Similarly, DNA gyrase can change, RNA polymerase can change, and 15S or 30S cannot recognize the target molecules. Yes, yeah, cannot, for example, cannot bind to the 50S subunit of the ribosome or 30S. Yeah, and bacteria can have ability to develop multi-drug pumps. What does it mean? So for antimicrobial action, these antibiotics should go inside the cells, yeah? And bacteria are very smart. They have developed pumps to pump out antibiotics outside the cell. These are multi-drug resistant pumps. Yeah, so any type of antibiotic that enters the cell then is pumped out by the bacterial cell. Yeah, by these multi-drug resistant pumps. This is another mechanism of action. So bacteria, this is not specific to antibiotics. While modification of transpeptidase confers resistance to beta-lactams or vancomycin, for example. A change in 50 subunit confers resistance to macrolized clindamycin or chloramphenicol, yeah? But cell wall synthesis blocking antibiotics still can work, yeah? So, but multi-drug resistant pump can pump out many types of antibiotics, yeah? And so this antibiotic development of antibiotic resistance is very important. And some and that's why we have to use antibiotics more rationally. We shouldn't prescribe antibiotics when there is a viral infection. For example, many doctors in Georgia liked, especially in the past, liked to prescribe antibiotics when you had fever, running nose, sore throat, and in many cases, these were viral infections, yeah, that did not require antibiotics. And when you use antibiotics, when you get a drink or antibiotics, then inside your body develops this antibiotic resistant flora, antibiotic resistant bacteria develop. Yeah, and when something happens, and again, when we go back to opportunistic infections, yeah, so in microflora, you might have antibiotic resistant bacteria, and it happens that you have immune suppression for any reason, and then these bacteria start to cause the disease, yeah? Then it's a problem to treat those bacteria, of course. Also, the proper dosage should be selected. Proper duration of the treatment, yeah, should be chosen for each medication. And also, antibiotics were used in farms to increase growth of the farm animals. Yeah, for example, cattle or chicken. And of course, we were getting those antibiotics when consuming pork or beef or chicken, and it also helps development of resistance in the pathogenic bacteria as well as in opportunistic ones. Yeah. So when then this resistance spreads through plasmids, through we have learned last time, yeah, horizontal gene transfer, through plasmids, through conjugation, through transformation, through transduction, yes, so different ways. So this resistance can spread. But some bacteria are naturally resistant to antibiotics. So they are susceptible without this selection. Yeah. For example, Pseudomonas, Acinetobacter, Burkholderia. There are several bacteria that have they're intrinsically resistant to bacteria naturally. Yeah, without human help to develop the resistance, and it's a problem to treat those infections. So. Does it make sense, or do you have any questions about this topic? No? Okay, let's continue. Now MCQs. Yeah. All of the following are modes of action of antiviral drugs except inhibition of protein synthesis at 70S ribosomes, inhibition of DNA synthesis, inhibition of RNA synthesis, inhibition of uncoating, none of the above. So, what do you think? Hussein says D, Ali A, Alexia A. Okay, 50S and 30S subunits. So together, yes, but together they form 70S sedimentation coefficient. So A is correct. So it was a little bit confusing for you, but together these four subunits have 70S. So A is correct. D is actually D is the wrong answer here. Yeah, inhibition of uncoating. On the previous slide, you did not see anything regarding uncoating, and uncoating is the stage in replication of viruses, not bacteria, when they remove their potential. We will discuss this when talking about viruses. Okay, very good. So inhibition of uncoating here is the answer. An antimicrobial agent should meet all of the following criteria except selective toxicity, the production of hypersensitivities, a narrow spectrum of activity, no production of drug resistance, none of the above. B, Alexia, thank you. What about others? B, Ali. Yes, that's correct. Why should we need the medication that produces hypersensitivity? Yeah. So it should have, of course, selective toxicity, narrow spectrum of activity. It depends what we want. Sometimes we need antibiotics that have broad spectrum. Yeah. No production of drug resistance. Yes. Now, so the most selective antimicrobial activity will be exhibited by a drug that inhibits cell synthesis, inhibits protein synthesis, injures the plasma membrane, inhibits nucleic acid synthesis, all of the above. Alexia says it's A, thank you. Ali Hussein, what do you think? Ali thinks it's A. Why do you think it's A? Why cell wall synthesis, blockage of cell wall synthesis can be can determine selective antimicrobial activity? Yes, protein is general. Yes, protein is general. Plasma membrane is the same. Nucleic acid synthesis is very same. Yeah, but in cell wall we have two major groups. Can be gram positives, gram negatives. Even more, we have acid fast bacteria that have mycobacteria, for example, that have many lipids and vaccines in their cell wall. So they can differ, yeah, in cell wall. So antimicrobials can be selective. Some antimicrobial can be effective only against gram positives, for example, and not against gram negatives, and may not affect mycobacteria, these acid-fast bacteria. And actually, we have just a few antimicrobials that are effective against these acid-fast bacteria due to specific cell wall structure. Yeah, you are right. Thank you. Okay. Next. So antibiotics that inhibit translation have side effects because all cells have proteins. Only in the few cells that make, yeah, only the few cells that make proteins. Because eukaryotic cells have ATS and there are 70S ribosomes in eukaryotic cells. None of the above, yeah? Why? So antibiotics that inhibit translation should be toxic to us as well. Okay, because all cells have proteins. All cells have proteins, but we have 80S and the bacteria have 70S. Yeah, so it can be different. Yeah. These antibiotics may not bind to ATS. They don't bind to ATS ribosomes. They bind to 70S. D, yes, because we also have 70S. And can you guess which cell organelle has 70S ribosomes? Eukaryotic cells also have 70S in one type of organelle. These are mitochondria that originate, yes, thank you, yeah, mitochondria that are actually prokaryotes, originated from prokaryotes. So these antibiotics can be toxic to mitochondria. Okay. This is very simple. Chloramphenicol binds to the 50S portion of a ribosome, which will interfere with transcription in prokaryotic cells, transcription in eukaryotic cells, translation in prokaryotic cells, translation in eukaryotic cells, DNA synthesis. The translation in eukaryotic cells. We're not talking about side effects here, yeah? Please don't confuse. What is actually the mechanism of action, yeah? Chloramphenicol binds to the 50S portion. Actually, chloramphenicol is quite toxic antibiotic. So for humans as well. So it interferes, of course, with translation, yeah? C, yes, Ali, thank you. So translation in prokaryotic cells. Yes, it was C. Okay. And I wanted to show you how do we test the susceptibility of the microbes, how it is tested. One of the methods. There are also different other methods as well, but what we do also in our laboratory, this is Petri dish, yeah, and we strip bacteria on their solid culture medium. So we'll cover the whole solid agar medium with the bacteria, and we put different types of antibiotics. These are papers, filter papers that contain different types of antibiotics. Yeah, A can be ciprofloxacin, for example. B can be trimethoprim sulfametaxazole. C can be penicillin. Yeah, and then we incubate this at growth temperature can be 37 for 24 hours so that bacteria start to grow and cover the solid agar. This is already grown, you see the bacteria, this turbid thing, what you see, this, yeah? This is bacterial growth, but you see the antibiotics have inhibited the growth of bacteria around them. You see here, and what we do in the laboratory, we measure this inhibition zone, and then we determine this bacterium is, for example, is resistant to that antibiotic. You see the inhibition zone is very small, but this is big. One should measure, and there's a special table. Yeah, for example, saying that penicillin, so this bacterium, Staphylococcus, for example, is sensitive, is considered sensitive to penicillin if inhibition zone is 30 millimeters, for example. Yeah, if we have narrow inhibition zone, it means that we have either intermediate sensitivity or resistance. Yeah, so we do this in our laboratory as well, and it's widely used. There's also e-tests, modified tests, but this is classic disk diffusion, Kirby-Bauer disk diffusion test. Does it make sense? Okay, let's move to the next slide. Yeah, let's discuss our last topics. Yeah, so we have here what is so how do you understand what is the sterilization? What is difference between sterilization and disinfection? No, we're talking about sensitization. Sterilization is sterilization methods, disinfection. Yes, Hussein, thank you. Sterilization is to destroy and remove microorganisms completely, including spores. Yes, Hussein, thank you. So we kill everything, yeah? So there are cases when we need complete sterilization. For example, of medical instruments, you in dentistry as well, you should sterilize your instruments, yeah? So to kill everything because your color, you may cause damage, you're removing teeth, you're doing some invasive activities, yeah? So in this case, you need everything to be sterilized. Yeah. What is disinfection? Yes, to kill. Yes, in disinfection, some viruses and spores have resistance. Yeah, but we still use disinfection. Why? Because we lower the dose, yeah, of microorganisms so that disease will not develop. So we disinfect the rooms in hospitals. Yeah, we do this disinfection. We can also make disinfection of some instruments that are not used for invasive techniques. Yeah, for example, some instruments that are used for breathing, yeah, nebulizers, for example, they don't need sterilization, yeah. And in this case, we reduce significantly the number of microorganisms. So you know that, you may know already that certain dose of microorganisms, so certain number of pathogenic bacteria are needed to cause the disease. Yeah, for example, in Vibrio cholerae, yeah, the bacteria that causes cholera, a million bacteria are needed to cause the disease. Yeah, in other cases, can be thousand initially here coli pathogenic strains, for example. So in disinfection, we lower the dose, the numbers of bacteria significantly, so that bacteria may no longer be able to cause the disease. But of course, the disinfection is not used for these medical instruments that we use for surgery or something. In that case, we need sterilization. Disinfection is for surfaces, for rooms, yeah, for some equipment, yeah, it's important. Okay, which disinfectants are called antiseptics? Antiseptics, thank you, are not that, just a second, prevent the growth of disease. Yes, they grow, but disinfectants in general, yeah, also prevent growth of bacteria. They kill also bacteria or viruses or sterilizants, spores as well. But antiseptics are those disinfectants that are used on living tissues. Yeah, for example, 70% alcohol, iodine solution. Yeah, yes. Not all disinfectants can be applied on the living tissue. We cannot use formalin, for example, or glutaraldehyde that are toxic, yeah, to living tissue. We use those disinfectants that are not toxic, and those disinfectants that can be used to clean the wound, to clean the skin. Those are named antiseptics. Yeah? So, antiseptics, they're not, it's not a sterilization. Again, we have disinfection here. We remove a significant number of microorganisms to prevent development of infection. Yeah? For example, if there is a cut, yeah, or if there's a wound, yeah, we wash and we treat the wound with iodine solution or with the hydrogen peroxide, yeah, 3% hydrogen peroxide. What we have used to prevent coronavirus, for example, use the disinfectants. Yeah, it was just, of course, we use antiseptics here. Yeah, so alcohol, hydrogen peroxide was used widely. Yes, 70% alcohol. Yeah, we were rubbing our hands. Yeah. But on the wounds, we can apply iodine solution, brilliant green, or something. Yeah, well, there are a number of different antiseptics that can be used. So, and we have physical methods, chemical methods of either sterilization or disinfection. So which physical methods do you know in general? Radiation, yes. Thank you, Hussein. We have radiation. Heat, yes. Thank you. For example, in your dentistry clinics, you will have heat ovens, for example, filtration. Hussein, thank you. Yes, you will have autoclave as well. You will have this heat oven that reaches 170 degrees in heat. You can sterilize glass. Uh, Materials. I don't know, in autoclave also can autoclave glass as well as some plastic materials that resist 121 degrees. Yeah, so instruments, metal, so different. You will have different sterilization techniques in your practice. Yeah, heat sterilization, UV radiation and gamma radiation. UV radiation is not so effective, but still we have in our laboratory UV lamps in our room. Gamma radiation is used to sterilize plasticware, for example, sterile gloves, yeah, of medical use purposes, plastic things, yeah, some catheters or whatever, they are sterilized by gamma radiation, yeah. And filtration, yes. In which cases filtration is used? To sterilize the fluids. We can autoclave the fluids if they stand high temperatures. But there are some fluids Yes, filtration removes unwanted microbes because microbes got stuck on the filter surface and the fluid goes through it. But in which case we need filtration. Yeah, there is some fluids, for example, can be Ringer solution or something that cannot be autoclaved because some components in the solution can change and can degrade, can be destroyed by the heat, but we need that component. In this case, filtration is used. Yeah, in our lab we use very often filtration, especially in solutions that has glucose inside, because at high temperatures glucose gets crystallized and becomes yellow. So in this case we use the filtration. Okay, great. And so we should move further. And just briefly, what is the mechanism of action, yeah, of those disinfectants or sterilization agents? Yeah, they disrupt proteins, they disrupt nucleic acids or lipids, yeah, and so the microbes get destroyed. Yeah, just two slides left, and then we'll finish. Just one MCQ regarding the sterilization and disinfection. Regarding sterilization and disinfection, which one of the following is the most accurate state? 70% alcohol is a better antiseptic than iodine, so 70% alcohol should be used to disinfect the skin prior to drawing a blood culture rather than iodine. Disinfectants kill both bacterial cells and bacterial spores. During sterilization by autoclaving, the temperature must be raised above boiling in order to kill bacterial spores. Transmission of milk-borne diseases can be prevented by pasteurization, which kills both bacterial cells and spores. Ultraviolet light used in the operating room to disinfect the room kills bacteria primarily by causing oxidation of lipids in the cell membrane. So D. Pasteurization cannot kill the spores, Hussein, yeah? Pasteurization actually targets the spoilage bacteria, yeah, that are quite often present in milk, and reduces the number significantly, but cannot kill the spores. Yeah, pasteurization is when we heat for a short time and then cool the milk products very quickly, and this treatment kills those pathogens that can be present in milk, for example. C. Okay, Ali says it's A. A 70%. So, yeah, actually, iodine is the better than alcohol, 70% alcohol. So A is false. This you may not know, but I will explain. So iodine is considered better than 70% alcohol. So that's why iodine is used prior drawing a blood culture. Iodine is better, but very often 70% alcohol is used. prior drawing blood, yeah? So let's see, yeah. During sterilization by autoclave, the temperature must be raised above boiling in order to kill the spores. Yeah, that's true. And let's discuss briefly the diagnostic techniques, identification of microorganisms, and why do we do. I will not ask you to describe in detail those techniques, because if you will need in the future, you will learn those techniques by yourself, but without contact, without being in the laboratory, without doing it by with your hands, it's difficult to understand. Yeah, but let's focus on why do we need identification of microorganisms, and in general which techniques are used. Yeah. Um, okay, so why do we need to identify microorganisms? Why do we need to know which microorganism infects the patient? Why do we need to detect the microorganism in the patient? Yes, absolutely. We should, yeah, it will give us the hint, not hint, but will then decide how to treat. Yeah, with antibiotics, if it's bacteria, if it's a virus, we'll use other approach. Yeah, it is very, very important. If it's fungus, we'll use totally different approach. Yeah, that's why we need to identify microorganisms. Yeah. And here you see four major categories, how we identify. And I will describe very briefly, yeah, phenotypic, morphological, yeah. So we observe under the microscope, we have different types of staining techniques, yeah. You have learned gram staining, there's also acid fast staining. We can also stain bacteria fluorescently, yeah, so different types of stainings exist, and you can see morphology, yeah, so phenotypic identification is, or biochemical identification is relatively old technique, but still used, yeah, so, and it's important how to take the sample, yeah. The person who is doing this, it is very important to know how to take the swab specimen from throat, from nose, how to take rectal swab or whatever, yeah, because the result largely depends on this technique. Then transportation is important. We should not keep the specimen yeah, for a long time. Yeah, so it should get to the lab as soon as possible for processing. Yeah, and we can culture then the microorganisms on culture media, and we can then observe under the microscope. We can do some biochemical tests. Yeah, different bacteria can eat different amino acids or carbohydrates, yeah, or sugars, yeah. So some can eat glucose and lactose, and other bacteria, mannose and arabinos, for example. And based on these tests, we can differentiate. Oh, this is E. coli. Oh, that is Shigella. Oh, this is Staphylococcus, or this is coagulase positive, gram positive cocci. So this should be Staphylococcus aureus. You see how we can identify bacteria based on these are biochemical tests, yeah, to check which bacteria is eating what, yeah, so which metabolic activities are present in those bacteria, yeah, do they have what metabolic activities they have. And still it is done, of course, and there are new modern, these big machines, Vitec systems used by the big diagnostic laboratories in Georgia as well, and were very expensive. And it gives identification within 24 hours or 48 hours. And this based on biochemical tests. Sometimes antibiotic susceptibility is also used for identification, and these tests in Vitec systems are done in many multiple plastic plates. Yeah, many tests are done, and then Vitec system gives you answer. So this bacterium is that, yeah, based on biochemical tests. But we have also serologic tests, immunological tests, yeah. We can, without culturing of bacteria, check for antibodies that we have learned in immunology course, yeah. And indirectly guess that this patient is infected with this virus or bacteria. Immunologic methods are also used for identification, yeah, like we have learned this precipitation tests, yeah. So many tests of bacterial identification, ELISA and so on, are based on immunologic assays. Yeah, either we can detect antigen or we can detect antibody. Nowadays, very important are genotyping techniques. Yeah, you know, I have questions you asked about PCR. Yes, exactly. And we identify bacteria very often based on PCR techniques. We amplify 16S ribosomal RNA genes that are conserved within the species. And we can then, of course, sequence these 16S amplicons and confirm that, oh, yes, this is that bacteria. So genotyping is very, very important and more sensitive and specific tests. But of course, we should do, it's best if you do biochemistry and culturing in parallel with the genotyping. Usually, this is done, yeah, for complete identification. And sequencing is very often done, yeah, without any culture. Many bacteria are non-culturable. Only 1% of bacteria are culturable. And also, it's true, can be true for pathogens, especially after antibiotic treatment. Many bacteria can enter viable but non-culturable state. They are there in the body, but they are not cultured. They cannot be cultured. In this case, these genetic methods, PCR, sequencing, and so on, can be used for identification or for detection. Yeah, first we detect, yes, it is there, something, and of course we are detection is then is together with identification. So genotyping is very, very important, and it is done in a very short time. Yeah, and for viruses, it is especially important. ELISA techniques and PCR and genotyping. So genotyping is very, very important for bacteria as well. There are also subspecies typing of bacteria that is important for epidemiological studies, which strains cause the outbreaks and so on. And we have also proteomics technologies, identification of bacteria based on protein content, and multi-top is a very expensive instrument. It is described in the lecture. You can check the presentation. It uses ionization, and bacterial cell is disrupted, and micromolecules then fly to the detector in this machine, and different proteins, depending on their size and charge, they fly in this instrument at different speed and reach the detector at different time points. And this pattern is then compared with the database, and then the bacterium is identified. Yeah, it is very also very expensive instrument, and it needs a very short time for identification. Yeah, very often we can't wait for culturing for 2 or 3 days for identification, and we need identification in a short time. Yeah, in this case, of course, these biologic methods and genetic methods or proteomics methods are very useful. Okay, do you have any questions? Okay, great. So thank you again for joining this tutorial. And so have a nice week. Thank you, Hussein, Ali, Alexia. Second Ali was also here, mistaken. Yeah. And Sarah was here. Okay, and have a nice week and meet you in a week. Bye bye.